This is a video to explain how to do thin layer chromatography, also known as TLC. The first step is to prep the TLC plate. The baseline should be drawn about 1 to 2 centimeters from the bottom of the plate, and the sample spots should be placed on the baseline. The spots shouldn't be placed too close to each other or too close to the edge of the plate, or they will run off the plate or interfere with each other. If the TLC is for a chemical sample, a TLC spotter, or glass capillary tube, should be used to spot a tiny amount of the sample onto the plate. The spot does not have to be large. In fact, if it's too big, then it can distort the results. In this example, the TLC samples came from marker ink. We spotted 3 different marker colors: blue, orange, and red. Once the TLC plate has been spotted, the TLC chamber should be set up. The solvent should be selected based on its intermolecular interactions with the samples. This is usually based on polarity, so that the polar solvent will interact more with the polar samples and show a difference between polar and nonpolar samples, or vice versa. There should only be enough solvent so that it soaks the bottom of the TLC plate, but does not submerge the spots. If the spots are under the solvent, the samples will just dissolve into the solvent and there will be no TLC results. A piece of filter paper should be placed inside the TLC chamber and soaked in the solvent. This saturates the inside of this chamber with solvent vapor and prevents the solvent from evaporating too quickly. In this example, the solvent used was 100% ethyl acetate. Now the plate should be placed inside the chamber so that the bottom is touching the solvent, but the plate is not touching the filter paper. The capillary action will bring the solvent up the plate and hopefully move the samples. The solvent front should not be allowed to move too far up the plate before the plate is removed. About 3/4 of the way up the plate is a good place to stop. In this example, we allowed the solvent front to move 3.7 centimeters up from the baseline, which took about 5 minutes. The plate should be removed carefully, and the solvent front should be marked with a pencil before it evaporates. A good tip is to line up the bottom of the tongs with the edge of the solvent front so it's easy to see where the solvent front was even if it does evaporate. The data collected from TLC is based on the movement of the samples in relation to the baseline and the solvent front. Sometimes the data isn't immediately visible, so the plate needs to be stained to see the samples. The stain will depend on the type of samples. In this example, since the marker ink is clearly visible, we did not need to use a stain to see the samples. Collecting TLC data involves measuring the distance between the baseline and the middle of the sample spot. If the sample spot looks more like a streak, just measure to the middle of the streak. The retention factor for each of the samples can be calculated by dividing the sample distance by the solvent front distance. In this example, the red dot moved 2.5 centimeters up from the baseline, so the red dot's RF value is 0.676. The blue dot moved 2.2 centimeters up from the baseline, so its RF value is 0.595. Since the orange ink ended up looking more like a streak than a spot, we measured the distance from the middle of the streak to the baseline, which was 1.6 centimeters. When we divided that by the solvent front distance, the RF value for orange came out to 0.432. And that's pretty much it for TLC. Bye!